Hello and welcome to Poly's first user group meeting. Thank you so much for joining us and taking time out from your busy schedules, wherever you are joining us from in the world. We have speakers and attendees join us from all over the world, very different kind of attendees, very different kind of users, and all that we are, all that Poly is today is thanks to all of you. Uh, in this particular conversation, we will talk about what's new Poly. We will try to make the case for why molecular data needs to be fair, why it is important for machine learning that you have your molecular data fair, as close to fair, as machine readable as possible. And we will also show you how Poly, under the hood, makes molecular data fair. At the end, we will go through a very broad overview of the platform, about 5 to 6 minutes, but we'll show you all the parts of the platform in very, very brief. And hopefully, if you're attending our other conversations as well, would be good context for what you will hear later on in this particular user group meeting. With that said, let's get started. Hopefully you can see my slide deck now. Here's the title slide, What's New Poly? And as I said, we'll cover a lot of ground in this particular conversation. We start off with talking about the opportunity with machine learning. And I think this is, kind of playing to the choir here. We all more or less agree with it. We understand that AI and machine learning are a huge opportunity for a lot of different sectors, and definitely for drug discovery and all of life sciences R&D. So just one particular figure to set context. Again, one caveat that we will not, for the purpose of this conversation, not differentiate between AI and machine learning. Those differences are there, but we will not get into them. What I wanted to make the case by this particular slide, before I lose some of you, and because you believe that you don't do machine learning, is to talk about the idea that machine learning as a subject, as a moniker, as a name, is very wide. It covers a lot of different things, and a lot of these things, for example, you would not necessarily associate with machine learning out of the box, right? Like when you think of machine learning, we often end up thinking about deep neural networks or other such more complex tools and algorithms, but they're not all. That's not always the case. I think most of you would be using one or more of them. Either you know about it, or maybe you don't, and they're covered under another name, another layer. But essentially, you must be using machine learning. And also, a lot of what we talk about in this particular conversation is as true of machine learning as it is of non-machine learning big data approaches. And of those, there are many as well. So, please don't believe that you don't use machine learning, or is this something that is very alien to what you're doing. Unfortunately, the problem with machine learning, and we are not the first people to say it, and I would want you to guess who this person is who has put this, who said the following quote. The problem with machine learning is that it's very hard, and often not for the reasons that we often associate. It's not the algorithms that are the hardest. In fact, a lot of those algorithms have become productionized, have become simple enough that you can just get them out of the box and apply them. But the difficulty is often the lack of clean data. And here is a person that you have probably heard of, who has said this quote, and as the speaker refers to, we spend most of our time just cleaning the data sets. And that's an important message that we will come back to again and again. And I would want you to guess who this person is. If you haven't so far, here's the answer for you, so to say. So there's a small quiz contest in the middle of the conversation. As you would associate, Vasna Simhan, as the CEO of Novartis, has pretty much all the resources at his disposal, right? Has as many resources as possible. And this is a particular quote from an interview that he gave in Forbes, in which he talks about how, in a lot of cases, again, machine learning has made significant impact, especially I think in this article, he talks about how in imaging, that's a particular field where Novartis has seen a lot of great impact because of machine learning. But often the challenge, the stumbling block, is access to quote unquote clean data. And we will talk about further as to what clean data looks like. More specifically for this audience and for what we do, for all of you, when you talk about data, we're talking about multi-omics data, and through some of these conversations, through some of our conversations, we'll also make the case for why multi-omics data is important. But again, I'm sure a lot of you already believe that. If you look at the slide, the first chart is, of course, talking about the explosion in the amount of data that is out there. That's an often said argument. You've all heard that like so many times that you don't want me to tell that. You don't want me to say that all over again. But if you look at the chart below, that's a little bit different. So what in this particular publication, and we quote the publication below, what the authors have shown that often data is not reused as much, even when it is in some very structured repositories. For example, in this particular case, an EMBL repository. Often in even those cases, data often does not get reused. So it's put there, but it's almost never reanalyzed. And the way we think about it is that the data associated with the publication is often the truth, right? The publication itself is a persuasive piece of argumentation on the particular data set, but it's the data set which is the truth. And a lot of you want to reanalyze it, reinterpret it, see it for yourself, but it doesn't happen that often. An interesting factoid here, if you notice, is that multi-omics data, for some reason, and the authors don't explain it either, gets reused more often than others. And if I were to venture a guess, I would say that it's more valuable than others. But again, that's me, and that might not be true for you at all. And what we're trying to tell you, I guess, through these slides is that you need machine actionable data, first of all. And the other part being that FAIR is a great framework to think about machine actionable data. And you'll hear the term machine actionable data again multiple times in this conversation, as well as other conversations that you will have with us, as well as you will hear the term FAIR. And again, FAIR is a community effort. It's a very broad initiative and can be interpreted in many ways. So, just in the interest of making that clear, this is our particular interpretation of the word FAIR. And what we have on the screen again is a quote from a publication that was written about the application of FAIR to industry. Why should industry care about FAIR? And the authors make the argument that industry or biopharma leaders will care about FAIR because FAIR is the ticket to unleashing the power of machine learning. That unless you have FAIR data, you really cannot harness machine learning as well as you should. And that's the argument, I think, pretty much that Vas Narasimhan is also making in his own words. And that's what we really believe in. That's the stumbling block. That's the first step. And that's the problem to solve. With that, we will go into more specifics of the challenges that we feel are there with multi-omics data. Why is it not machine readable in the next few slides? The biggest challenge we feel with a lot of the data that is out there, and whether that be data in the public domain or data lying inside companies, is that the data schemas are inconsistent. Before we go further, let me just tell you that the examples that we will use in this section are all real. They have been taken from our work that we have done over the last few months on Geo, and that's one particular data repository, and that's an example that we are using because it's a publicly available resource. But that's an example that I think applies very much to even data sets that are stored in-house within your companies. And if you see the example on your left, that's an example where all of the data files in this particular study in Geo have been separated out into their own files. And of course, as most of you who would realize, it takes a fair bit of work to combine them for one particular study itself. And of course, the problem is that if you go to another study, whether that be within Geo or whether that's a study that a colleague of yours has done like 2 years ago, the way the file might be distributed might be completely different. So it's not predictable in any kind of easy way. Again, if you look at the example on the bottom left of your screen, that's an example in which gene identifiers are inconsistent. And again, this is a problem very common with Geo, as well as it's a lot of different data sets that we, for example, see from our customers, right? Whether that be genomic data in this case, and of course the problem is much larger if you're talking of metabolomic data or in general mass spec data sets. So that's sort of problem number one, and I think the biggest problem that is out there. The next part to this is that metadata is very, very seldom standardized. It's one of those things which I believe gets talked about very often, but is very rarely enforced, if at all, right? And again, a repository like Geo, which has some of these standards enforced to some extent, and depending upon where you are, your organization might have stricter standards, or more often not as many standards which are enforced. But as you would see, even just the way to describe replicates in these studies is very, very different. And then if you're analyzing data at scale, again, if you're looking at just one data set at a time, maybe this is something that you can deal with. Even then, I don't think this is where you want to spend your time. But if you're looking at just one study, maybe you can deal with this kind of challenge. But if you have to look at hundreds, this is completely sort of going to stop you, right? You're not going to be able to move forward. And if you look at the query that we have talked about, find all type 2 diabetes studies, this kind of study, this kind of query is, of course, very, very difficult if you don't have metadata standardized at the study level, right? So there is a sample level metadata standardization as well as at the study level, and you need both, right? You need both for different reasons, but very rarely, if ever, is metadata standardized the way it should be for machine learning or other big data approaches. The same data sets after verification through our platform, right? And again, this is a real example, as I will tell you again and again, on our platform. And what you see is, I think it's pretty obvious on the screen. The samples have been combined together into one particular format. And this particular format is, for example, consistent for all the geo data sets, all, about 45,000 that we have released on our platform. This is consistent for all of them. And this is what we can do for your data sets as well. So if your organization has a large variety of data sets where this kind of format is not consistent, we can verify it and put it in the same format, standardize the gene names or metabolite names or protein names, whatever that might be the case, as well as standardize the metadata using ontologies and using consistent vocabulary, right? Just the example that you see, and I'll focus on the yellow parts here. This is a part of the standardized vocabulary that our platform has. This is a kind of a vocabulary as well as an ontology, and there are slight differences between those two. Is something that we maintain. So it's something that we take care of. This is a real example where we have done that for some data sets from Geo. As you would see, my intention is to say that this is not easy, and for very good reasons we believe this is not easy. Again, none of you have to look at all of the 100,000 data sets and all of 50 omics repositories and all of the formats ever, right? That's not the point. I really hope that you don't have to, because our scientists and our team has struggled a lot to do a fraction of this. But you get my point. I mean, you want to look at as many data sets as possible, right? For machine learning, or just to have more confidence in your results, even if you're comparing your one particular experiment, and if you want to see that the same genes have come out in similar quote unquote experiments in the past, it can be very, very challenging if these things are not taken care of. And this is where automation and tech expertise, we believe, become really, really important. And even if you can do it for a few studies, that we're not sure that's the best use of your time. We really believe that scientists should be spending their time interpreting data, making formulating insights or making connections, the kind of things that machines cannot do. But data cleaning is not something where your most valuable resources and you yourself most probably should be spending a bulk of your time. Now we'll spend a few seconds looking at how exactly does Poly solve this problem, right? I think we have talked enough about the problem. How do we solve it? And primarily, if at a very high level, what we do is we automate data ingestion, and we'll go a little bit into talking about how we do it, but that's the primary message that I would like you to take and retain. And in this particular example, the infographic that you see on your slide, we have shown how we have done this through what we call connectors for a variety of repositories, public or otherwise available. And what that means is that the data lakes on Poly are up to date. And again, we'll talk about how up to date they are. We are increasingly working on making them more automated. But the idea being that we take data from these public repositories, our connectors pull that into our infrastructure in an automated fashion. They go through what we are calling, this particular diagram here, which is our curation infrastructure. It's a combination of software and models and other things. So it goes through our curation infrastructure and becomes normalized and present in a consistent tabular schema. Now, why is that important apart from the up-to-date part? It's important because let's say if you're writing your analytical code, right? Whether let's say you're doing something in a notebook, or whether you're making, let's say, a Shiny application to be able to give to your colleagues so that they can do something on any of these repositories. They can do that on Poly database. And what that means is that your application, your code is only talking to the Poly database. It's not talking to these repositories here. And therefore, even if something changes, if new data comes, of course, it's available. That's part one. Even if something changes, let's say in GTEX or more plausibly in Geo, that does not affect your upstream, your downstream code. So your code does not break even if a new field is added, for example, right? And so you can just focus on your code and leave the data engineering, the data ingestion to us. And at this point, we support a lot of public repositories. Again, happy to please reach out to us if you want to understand which repositories are already available. What is coming soon are connectors for internal data stores like LIM, CLN, other kind of processing software from Aspect or Single Cell if you're using them. And we'll talk more about them in our roadmap conversation. What our infrastructure does is that it verifies data faster and better. And for the faster part, as you would see here, we also do expert curation to generate training data sets. So these are training data sets are essentially annotated data sets on which our machine learning models learn, quote unquote, and work. So expert curation itself for us has been rising very steadily, and we'll talk more about how that has been done. But if you look at how automated curation has grown, it's absolutely exponential, right? And that's the idea with machine learning, or in general with automation, that you can use a little bit of training data sets to really exponentially increase the automated process. And that's what we had done for a lot of these repositories. And this graph, again, that you see on your left represents just how fast we are adding data sets to the platform. And we'll talk, we can tell you very clearly that it's much higher than many of our competitors are doing or have done. Now, while we add more data, of course, we have to ensure quality, and that's the better part, right? So this is a fair score that we have defined based on guidelines that are published, are in a peer-reviewed journal. And the guideline takes a bunch of parameters, about 10 or 11, and on each of those ranks a data set or a study, and then you kind of combine all of them to get a score. And what we have demonstrated is that the fair score, after going through our infrastructure, is demonstrably higher than what is there in Geo, more than 100% better. So not only is it faster, we are ensuring quality through a lot of checks and balances. And coming to the part that I referred to earlier, we use expert curation to create training data sets, and not just to create training data sets, but also for QC, right? So models, after they have worked, the data is also QC'd by human experts. This is again a real screenshot of our curation infrastructure, what we call our curation infrastructure, where expert curators create the training data sets or perform QC on the data sets as the need might be. And what this infrastructure has done is that it has allowed us to scale up human expert curators by a factor of 10x, right? So, and again, we can go into some detail maybe at a later point, but suffice it to understand that it goes from all the way from removing very simple logistical nightmare as to where do I find the next data set to curate, to, for example, the models, as the color represents, predict perturbation and control, and the human curators only have to kind of agree with that or differ. So it really scales up the human experts. And thanks to that, we have built one of the largest and some of the largest training data sets ever, and it's only increasing. Again, in our roadmap conversation, we'll talk about where we are going with this, but our intention very firmly is to create the biggest training data sets for multi-omics data that are out there to curate and clean data better than anybody else. Finally, and this is the point that we made earlier as well, we curate metadata and to make it consistent, to make it controlled using a vocabulary, using an ontology. And there are a couple of examples here just to show how our models very much agree with experts. Again, this is real. This is a real example. This is a real screenshot, as well as some more rigorous studies that we have done on two particular tasks. I won't go into these details here. If you're curious, do look out for our tech note. Our team has written very cool tech note, which I would definitely suggest all of you who are curious to learn more about this to go through. And what we demonstrate there is how our infrastructure, our machine learning, and more in general, our models, our infrastructure have improved upon not only the benchmarks which are published, and we use some of the most rigorous, most recently published benchmarks for these kind of tasks, but have also been almost equal to human experts, right? So that's the power, that's the secret sauce, if you might, that allows us to verify data faster and better than anybody else. Now, moving on, in this particular section, we will, as I said earlier as well, we will, in very brief, look through all of the platform, right? I've told you what the problem is, why is it important, how are we solving it, but now, what does it mean for you? How do you use it? How does it look on the platform? So a very broad, very, I wouldn't say in-depth, but covers and touches all the parts of the platform. I will touch all the parts of the platform in this particular video demonstration. We start this video demonstration with one of our newest parts of the platform. This is a pre-recorded video that one of my colleagues has made to demonstrate. This is our newest product, which we're calling Omics Wiki. As you would see, there are a bunch of curated data sets taken from Geo and available with a fully made dashboard, which is right out of the box useful. So if you want to look at any of these data sets, you can just get started with an Omics Wiki. And it can also be created for, let's say, hundreds of data sets at scale. Now we go to another part of the platform, the data lakes. And again, for this particular example, we will look at two different kinds of data lakes. This is just to give you a snapshot of what kind of data lakes are on the platform. As part of the UGM, we are also doing a big release of our COVID-19 data lake. This is a very good resource for COVID-19. And as you can see, it allows you to select based on your parameters of choice. So you can filter the data sets that you want to look at without having to go through all of them. We go to another one, and this is a multi-omics data set. You'll see that they are much smaller. Now, this is a curated data lake, which our customer solutions team has created for a fictional customer, right? So if you were a fictional customer, and if you came to us and said that this is the kind of data lake that we need, we can create such a curated data lake for you. Not only do we help you create this, but also help you find the data lake. Now, using that, as you would see, with one click, we started a particular application. This is what we call a single cell application. It's a Shiny application that we provide, and you would see that because the data is curated, the user can do a bunch of things right out of the box. Again, this is a Shiny application that we provide for single cell data analysis. If you have one of your own, somebody in your team who is more familiar with R Shiny, who's more familiar with coding, can bring theirs here. Now, this is a different kind of application. I think a lot of you who work with single cell data would be familiar with it. It's called Cell by Gene. This is, again, something that can be hosted by your colleagues. Again, if you don't have those colleagues, our customer solutions team can also do that for you. But your colleagues can host the applications that you use today on Poly, and they can all be started from the data set, from the same data set. As you would see, now we have started a notebook with the same data set. So you just selected that data set. You started a Shiny application. You started Cell by Gene. You also could start a notebook and start analyzing it, right? It's the same data set, just one click. That is the power of curation, right? So we are not even talking about something super fancy ML here, but just being able to start one data set without any problems. This is what we call workspaces, and you would see, the operation that you need to manage your data, and more like files, for example, Excel or CSVs, your MZ files or other kind of files. You can upload very large files here and store them, create folders, whatnot, um, bring in data pretty easily. If you have large data sets as well, they can be brought on, and this is where your work can be stored. This is something that is shareable with your colleagues. Uh, we version your data sets. So, for example, if you have a file and there are many different versions of it, whether that is a notebook or whether that's a CSV, that is versioned and kept on the platform. What you see my colleague doing here is how you would select a machine of your choice for a particular notebook. And you can do this for notebook as well as you can do it for example, a mass spec processing software like LMaven, right? Sometimes you have a larger data sets, you need a bigger machine. And that's just how you can select a bigger machine and get started. Right at the end, and again, as I said, this is a brief brief overview of the platform. We'll show you our newest product, what we're calling Data Studio. This is a GUI-based data analysis, or I should say data analysis building platform, where your bioinformatician colleagues can create these kind of tools very easily using their components, their pieces of code, or the components that we provide. And as you would see the user here, they're doing different kinds of analysis. The differential expression component is version, so you know which one you're using. You can run tasks. These tasks can be small or can be very long as well. Again, we allow you to select a machine of your choice here. So some of your computations can be small, some of them can be very large. You get pretty interactive plots, and you add them all to dashboards, which is pretty interactive, as you would see. In this particular case, we are naming the report. We can kind of edit and modify it, just by clicking through, give it a name, call it whatever you want, put some images where, arrange it the way you like, and then share it with your colleagues. And you can share the dashboard, or you can come back and share your whole workspace, which is all of the work that you have done. So that's in very, very brief, all of the platform. Again, as I said, this is going to be quick. This is very quick. But for more details, of course, you can always listen to some of our other conversations or reach out to us. We are happy to show it to you any way you like. I hope you enjoyed the small demonstration, that quick glance, as we call it, of the platform. And the only thing left for me to do now as part of this conversation is to give a big shout out to some of the other people who are talking, our users, most importantly, as well as some of our experts who understand this much better than me, are giving some pretty interesting talks. Big shout out to them. Do attend those, and you can see a variety of use cases in which Poly is being used and how that's being done. Finally, I have referred to the roadmap conversation a few times, so I thought I should end with that. And this is at a very broad level what the roadmap looks like. I think we have a very exciting path forward, something that will appeal to a lot of you, and do join us for that. With that said, thank you very much. You can always find us at elucidator.io. We are available on the chatbot, on our platform. Otherwise, reach out to any of us. Thank you so much for joining us for this particular conversation. Thank you so much to all of my colleagues whose hard work made this possible. But most of all, thanks to the community, thanks to the users who have joined us for this particular conversation. We wouldn't be here without you at all. Thank you so much.